and today in Crash Course Molecular Biology, we will talk about xenotransplantation. Xenotransplantation is the process of grafting or transplanting live cells, tissues, or organs from one species to another. The goal of xenotransplantation is to provide a nearly unlimited supply of cells, tissues, and organs so that damaged organs such as hearts, kidneys, and livers may be replaced. Tissue transplants such as skin grafts and borrows may be given, and cell transplants may be used to replace a damaged cell in diseases such as diabetes and Parkinson's. The development of xenotransplantation is driven mostly by the demand for organs and clinical transplantations. Doctors first tried to transplant animal organs into humans over 100 years ago. In the early 1900s, attempts were made to replace failing human organs with animal organs from pigs, goats, lambs, and monkeys. Of course, all of those attempts failed. They would not know why until 1944 when Peter Medawar discovered that the recipient body undergoes an immune response due to the transplanted tissue or organ. These donor tissues are recognized by the immune system as foreign and are attacked, causing transplant rejection. Following this, immunosuppressive drugs were made in the early 1960s. The discovery allowed the success of human-to-human transplantation and raised the demand for donated cells, tissues, and organs. Xenotransplantation was again investigated to help solve the supply problem. In 1963, Dr. Thomas Starzl made the first successful attempt at xenotransplantation. Kidneys from baboons were transplanted into 6 humans. The patients survived for 19 to 98 days. More organ transplants, such as hearts, kidneys, and livers, were attempted in the following years. However, success rates were still low compared to the human-to-human transplants. Cell xenotransplants are considered the most promising due to their use in the treatment of disease and the relative ease compared to organs and protecting them from the recipient's immune system. In 1996, pig cell transplant was conducted to treat type 1 diabetes of the patient, Michael Hellier. Researchers also started investigating the use of genetic modification to create transgenic pigs and prevent transplant rejection. All xenotransplants was however banned worldwide in 1997 when concerns about the pig virus, porcine endogenous retrovirus or PERV were high. Recently, bans are being lifted on a case-by-case basis, and in Russia, New Zealand, and Argentina, clinical trials on the use of pig cells to treat type 1 diabetes is again underway. The issues faced by xenotransplantation are transplant rejection, cross-species infection, and ethics. There are 3 types of transplant rejection: hyperacute rejection, acute rejection, and chronic rejection. Hyperacute rejection occurs rapidly, sometimes even before the operation is complete. Acute rejection occurs in the first few weeks or months after surgery. Chronic rejection occurs over several years. Several approaches are being investigated to combat transplant rejection. First is immunosuppression, or the suppression of the recipient's immune system. This may be done with drugs. However, it can be toxic and decrease the recipient's ability to fight off infections. Immunosuppression may also be done by removing the antibodies of the xenotransplant. The second approach to combat transplant rejection is genetic modification. Here, donor animals are modified so that their tissues will no longer be recognized as foreign. Products of this process are called transgenic animals. The third approach is only possible for cells and involves the protection of cells from the immune system. Transplanted cells are coated with a substance that prevents them from being recognized by the recipient's immune system. An example of this is the technology being created by Living Cell Technologies in New Zealand. LCT uses microencapsulation with a seaweed-based coating to protect pig cells. Cross-species infection is tackled in choosing the donor species. Concordant species are genetically closely related species, like mice and rats. Discordant species, on the other hand, are not closely related. For example, pig and mice. Based on experimental records, concordant species are more compatible than discordant species. Non-human primates such as chimpanzees and baboons were used in early experiments of animal-to-human xenotransplantation. However, concerns spread due to the risk of spreading infectious diseases such as herpes. Chimpanzees were also listed as endangered in the 1960s. These factors caused a shift from using primates to using pigs. Pigs are easy to breed, produce large litters, and have organs similar in size to humans. There is also a lower risk of contracting infectious diseases from pigs. Fewer ethical concerns were raised due to the public acceptance of killing pigs for food. The main ethical issue being raised by ethical committees is the issue of health. Diseases such as AIDS or acquired immune deficiency syndrome have been acquired by humans from animals. All pigs carry PERV. Although this has not been shown to infect recipients, it could possibly cause a new epidemic. Other ethical issues are the welfare of the pig subjects, the religious traditions involving pigs, and the unpleasant idea of crossing the boundary between human and other animals. To address these issues, the World Health Organization, or WHO, has held events and released guidelines on performing xenotransplantation clinical trials, the latest being the second WHO Global Consultation on Regulatory Requirements for Xenotransplantation Clinical Trials, held in October 2011 at Geneva.